Excellent. Okay. Well, I've been at Manchester for quite a long time now, about 25 years. And before that, I worked at the Institute of Zoology in London for around about 15 to 18 years. And before that, I was up at the University of Edinburgh, where I did my PhD and then stayed on and joined the staff in the university for a short while before moving to London. And I've also worked in the States and in Australia, and I've been involved in a few projects in China as well. So I've hopped around a bit, but I've developed a very strong interest in biological clocks and timing that arose originally from my interest in seasonal breeding in animals, and from that, an interest in the molecular mechanisms that time rhythmic behavior. So what I thought I'd do today is tell you a little bit about the circadian clock and human health. And what I've done is kind of develop a hybrid lecture, because I put some real data in, like I was giving a university seminar, and some cartoon summary slides to mix it up around a bit. And it's become a very large subject over the course of the last 15 to 20 years, absolutely enormous. There's a huge literature now in this field. And as some of you will know, 3 colleagues of mine were awarded the Nobel Prize in Medicine in, I think, 2019 for their pioneering work in the discovery of the underlying molecular mechanisms driving circadian rhythms and biology. So what I'm going to do today is to give you a very brief overview with 2 or 3 slides that I showed at the last talk, and then introduce you to a topic which you may not be that familiar with, which is the crosstalk between the timing systems in our body and those in animals and the immune system. And I'm going to tell you a little bit about immunity and a little bit about metabolism and how we think those 2 processes interact through the activity of the clock. And I'll finish it with some general remarks about circadian medicine. So to start the ball rolling, we live on a rhythmic planet, which is tilted, and that tilt gives us the seasons. And the rhythm of the daily rhythms, of course, arise from the rotation of the Earth. And we've now known for really a very long time that pretty well all organisms on the planet, from microorganisms up to complex vertebrates, show endogenous circadian rhythmic behavior. By which I mean that if you keep any particular organism of interest under constant controlled conditions and measure appropriate processes, you can identify free-running cellular autonomous rhythmic activity in systems, and in some organisms like cyanobacteria, pretty well the entire genome is under circadian control. I don't think there are any genes in cyanobacteria that aren't under the control of a clock or a timer. And this process is driven by clock genes, which have been identified in all major organism groups now, and that's kind of history. What you see is what you get. And at the bottom of that little cartoon there, from a review by a couple of colleagues of mine, you can see some of the mammalian clock genes, which show very close homology to those that we see driving the behavior of insects, such as Drosophila. The Drosophila clock is remarkably similar to the human clock in terms of how it works. I'm not going to go into clock genetics or the biochemistry of it. It's a separate topic, and it's a bit specialist. But we'll mention just, I think, one gene as we go forwards. And circadian clocks, we now know, have been around, or we can identify them right back to the beginning of prokaryotic evolution. So we're looking at processes which are at least 2.5 billion years old. They're some of the most ancient regulatory systems that we find in living organisms. They may even anchor back, and we don't know, to about 3.5 billion years ago, which is when we think the first prokaryotes appeared. Not terribly far short, only by about 1 billion years of the origin of our solar system. And fungal clock genes and plant clock genes have been around a lot longer, as you can see on that little cartoon there, than animal clock genes. And the animal clock genes that include vertebrates and invertebrates show really quite remarkable similarity in terms of how they operate. But the fungal and plant clock genes are really quite different beasts. And in terms of humans, our own clock, as we know, drives multiple processes, and I'll come to some of those a bit later. But here's a very, very old piece of data from Jürgen Aschoff, a very distinguished German physiologist who's now no longer with us, who was really a pioneer in the field of studying human circadian rhythms. And this is sleep-wake cycle of a medical student, volunteer medical student, and at the top this individual's in a normal light dark cycle, and the solid blue lines are when the person's asleep, and the dotted lines when the person is awake, and the blue triangles are when the person has the lowest body temperature in the rhythmic body temperature cycle, which occurs towards the end of the sleep phase. And then on day 10, this person was put into constant conditions in isolation, with no external access to a light-dark cycle. And you can see that this person's sleep-wake cycle is now running free and a bit longer than the natural period of 24 hours. And then at the bottom, you can see this person comes back into the light-dark cycle and re-entrains within a matter of about 4 or 5 days to a normal pattern of activity, and the sleep-wake cycle is now locked onto the daily solar cycle. So, and this happens, although we're unconscious of it, every single day. We're given a little nudge by light. Light is the primary entraining force here, and that resets our clockwork and brings us back into synchrony with the real world. And that statement's true for pretty well all organisms, with a few exceptions here and there. Light is the predominant force on our planet, and there's a fascinating story to do with the evolution of clocks and the evolution of photoreceptors in different cells and cell types, because they've clearly co-evolved across time. So in humans or in animals, vertebrates, the search back in the early 70s, late 60s, early 70s, was to try and identify anatomical structures that might be providing a sense of circadian time. And two separate groups simultaneously in 1972 published papers showing that there's a structure in the bottom of the hypothalamus, which is shown on the right-hand side there in sagittal section, called the suprachiasmatic nucleus. And it's anatomically linked to the eyes via photoreceptors that reside within the retina, and their nerve terminals, the terminal fields, are in the suprachiasmatic nucleus, which is in the hypothalamus. And that's a really interesting strategic place to be if you want to control body physiology, because outflow from that area controls all sorts of other circuits, including the sympathetic and the parasympathetic system, and also much of the endocrine system that can be wired back through various links to the suprachiasmatic nucleus. So that's it there. And on the left-hand side, you can see the underside of a human brain. And the suprachiasmatic nucleus, I don't know if you can see my pointer. Is that possible? Can you see the pointer?
Yes, we can see it.
You can see the pointer. Okay. So the suprachiasmatic nucleus lies immediately above that structure. This is the bottom of the brain here. Those are the nerve terminal fields. So that's where the clock is in the brain. And for a long time, it was thought that that was the so-called master clock that drove everything else in the body via outputs from the central nervous system, linked in some magical way to every cell in the body. We now know it's much more complicated than that, and that discovery there was made by a Swiss colleague, a Swiss biologist, who noted that there were strong circadian rhythms in cultured cells. And he then went on to show that that was true for most, in fact, all of the major body organs of the body. He was working initially on mice. And we can now say that pretty well every cell in our body and in any mammalian body has got self-autonomous clocks. And if you take those cells out of the, I see someone's in the waiting room, I'll just admit them. If you take those cells out and culture them, they'll continue to oscillate with robust 24-hour rhythms. So what's happening is that the brain clock linked to the outside world and the eyes is in some way synchronizing all of these peripheral clocks, which are ticking away in phase and in synchrony with that of the central nervous system. And I'm going to explain a bit later on as to how we think that actually might work, but I won't go into the details of that right now. But if we look on the right-hand side, there's a little collection of activities, physiological processes, which are under clock control. And I could just as easily have filled this entire slide with boxes with other sorts of physiology, because I'm unaware of any major physiological system in the body that is not under circadian control. And I'd buy a reasonable bottle of wine for anybody who can firmly identify for me any major physiological circuit which is not under circadian control. So there's your challenge for you. And this whole process is driven really quite remarkably by about 15 to 20 genes that act as repressors and activators and oscillate over a natural period of 24 hours. It's an extraordinary piece of biochemistry. It's temperature controlled, it's temperature compensated rather. And this clock runs with remarkable precision in cultured cells in the laboratory. And we've done quite a lot of work in that area, and it's an area of continuous interest to us. So that's the circadian clock. It's a regulator of synchronized physiology throughout the body, and every major physiological system is under circadian control. And here's a little diagram on the right of some of the circadian circuits that are involved in this process. Period and cryptochrome, which act as repressors, and clock and BMAL, which act as activators. And the mechanism of the clock works through rhythmic feedback repression through the purr and cry proteins. And those are the systems that regulate, we think, circadian rhythms throughout the body. If you use modern genetics, you can screen for every single gene in a tissue sample or a cell sample using techniques such as RNA-seq, which have become remarkably easy and efficient to use and relatively cheap by comparison to the early days. And this has allowed people to look at the pattern of gene expression over the course of the circadian cycle. And what you see there on the right, the bottom, is an example of two tissues, the liver and the lung, and I'll tell you a lot more about the lung in a minute, in which samples were taken from cohort samples are taken from different mice at different times of the circadian cycle. These are free-running mice in continuous circadian conditions of darkness, no exposure to light. And you can see that the number of genes that are expressed in a strong rhythmic pattern varies across the circadian profile, with most genes being expressed strongly in the liver late at night. And that kind of fits because the mouse, of course, is nocturnal and starts eating at around about CT12 on that diagram, the shaded area. And many of the genes in the liver that are under circadian control are involved in metabolism, metabolic processing, and xenobiotic detoxification. And the total numbers of genes here, there isn't a scale on that, run into the many thousands for each of those two tissues. The lung is slightly different. The peak phase of activity is at about circadian time 12, when they're around about, if I remember rightly, 2,500 genes at that time point that peak at that phase. And you can see that these two tissues are really highly rhythmic, and there are literally thousands of genes that are linked back to this clock and under clock control. So I want to talk today a little bit about the immune system and one or two things that we've learned recently. And this is from a review we wrote a few years back now, which just summarizes in a very simplistic way a range of different immunological functions which are known to be under circadian control, and where circadian disruption has implications for, in particular, human health. So I'm not going to run through each of those boxes, but you can get an impression there that a very large proportion of the immune system is indeed under circadian control. And there are a large number of publications, some of which we've generated and others by other colleagues. And this is from a review we wrote a few years back, which shows the current state of knowledge then of all of the different major cell types of the immune system, or many of them, which have been shown to be under rhythmic control and have rhythmic oscillators running within them that regulate the physiology of these particular cell types. Furthermore, we find that the core clock, which is shown here again, you've seen bits of that already, rather remarkably, some of these elements that drive the inner circadian clock are also actually pretty critical in terms of driving the lineage of developing cells that go on to become immune cells. And in particular, the ROR proteins at the bottom right there, which are key regulators of clock and BMAL, those are also important in the Th17 lineage cells of the immune system. And at the top of the diagram, there's another little circuit involving NIFL3, which is part of another feedback loop that's core to the clock. But if you knock that gene out, you end up with a mouse that's got no natural killer cells and is in some trouble. So as we explore the biology of the circadian clock and look using genetics at what happens when we perturb various genes, we find that we have unexpected effects primarily on the immune system. So the immune system is very, very closely interlocked to the circadian clockwork mechanism itself. So I'm going to tell you a little bit about inflammatory diseases, and I'll tell you something about some work on mice and something about work on humans. The mice, of course, give us the great advantage that we can do genetics and experiments that we can't easily contemplate in humans, but with increasing technologies, we can now carry a lot of this work into humans and even take the work further forwards in humans than we would bother to do in mice. So I'll give you a bit of a flavor as to how that works. So in terms of human inflammation and inflammatory diseases generally, they've long been known to be frequently under circadian control, and there's a particularly well-established phenomenon for rheumatoid arthritis, and that's what you're seeing here. These are the patterns of joint stiffness on the left-hand side in red, and pain and functional disability in a cohort of patients, and this is from a publication by Cutolo and colleagues. And on the right-hand side, you can see a marker for what we think is a key protein involved in this, which is under the control of the clock, and it's a protein called interleukin 6. It's one of the cytokine families. And you can see that in the healthy subject, it's got a natural rhythm with a relatively low amplitude, but in the RA patients, the amplitude is almost doubled. And we think actually that's now telling us quite a lot about the process of the disease and how that disease process is interlocked to the circadian system. And for a long time, it was thought that RA joint stiffness was associated with time of day. You can see that it peaks late at night, in the middle of the night, because people were just lying flat and not moving around and getting active. But a number of very clever control studies have been done to correct for that. And it's basically an underlying circadian mechanism that's driving this, and nothing to do with the rest cycle per se. So moving on, I'm going to just tell you a little brief story to do with the lung and lung inflammation, and then I'll tell you a little bit, I'll mention some fascinating recent work on vaccination, and I'll even show you one slide from a paper that's just come out. It hasn't even been, it's available online, which is an extraordinary story to do with COVID vaccination and the efficacy of the circadian clock in regulating how that works. So bear with me. So inflammatory diseases generally are highly prevalent in humans, and the lung is no exception. And inflammatory lung disease underpins all sorts of pathologies, many of which are severely life-shortening. And we got interested in this in mice because we thought that we could use combination of genetics and experiments in mice to try and unravel some of the core mechanisms that might underpin this. And early on, I haven't put the citation to this, but we published this back in about 10 years ago now, 10, 15 years ago. We identified a population of cells that lined the respiratory bronchioles, which are these very fine structures here at the end of the network of bronchial structures that then leads to the gaseous exchange zone, the alveolus, these very thin-walled cells here that absorb oxygen. And the cells that line the bronchioles have been quite well categorized. And there's one particular cell type that's non-ciliated. It's called the club cell, which is shown here. And we were able to show by a variety of methods that these cells were particularly strongly rhythmic. Here's some real data. These are isolated cells in culture, and you can see them oscillating. We've got a gene that emits light linked to the clock, so we can culture these cells from transgenic mice, and you can see the lovely oscillation in those genes. And our attention was really focused on those cell types as a possible candidate for how the rhythmic physiology of the lung may operate. And the model that we used was to apply endotoxins such as lipopolysaccharide, which mimics gram-negative bacteria. It's a cell wall protein. And in a variety of studies also later on, we've used bacteria, viruses, and even parasites. And I don't have time to go into all of those studies in the lung and in other body tissues. And one of the ways in which the immune system reacts to a foreign body, such as a bacterial infection on the airway side, which is shown at the top here of the diagram. So these will be the bronchial structures here, is to trigger a local immune response involving resident immune cells such as macrophages, which send out signals to the bone marrow, which then triggers the release of neutrophils, which are very short-lived cells and very aggressive, and they punch their way through the endothelium into the tissue, into blood, and find their way to the target of the infection, where they engulf the invading organisms. And if there are people who'd like more detail of this at the end, I'm happy to provide it. And if there are immunologists taking part in this meeting, apologies for the terrible short circuitry I've indulged in describing the biology of these fascinating cells. But they're attracted to the target site by chemoattractants, cytokines and chemokines, which tell the neutrophils where the infection is occurring. And if you do an experiment in mice in which you administer bacterial challenges or lipopolysaccharide at different times of the day, you trigger, and I'll show you some data in the next slide, you trigger a marked neutrophilic response, which you can recover from the lung. You can measure the amount of neutrophils relatively easily. And this shows a little cartoon here in which you've got a a bacterial challenge. And then here are the resident macrophage cells that have been sitting there. They're long-lived cells. They will have been there throughout development in the lung. And they will trigger a response, which will then call on a neutrophilic response, which is delivered through the bloodstream. And here the neutrophils fighting their way through to deal with the infection. And I should say that one of the primary target cells that COVID attacks are these very bronchial epithelial cells. And the primary cause of death in many patients is associated with the strong reaction of the immune system associated with neutrophilic invasion, trying to deal with this overwhelming infection in these epithelial cells. I also should point out that we started working on this problem at least 10 years before COVID ever appeared. And I'm certainly no coronavirus expert. I know nothing about it really, but I will tell you what I do know about the immune system of the lung and how these cells are involved. So here's some data from an experiment that Julie Gibbs in our group undertook. And what we did is we took mice and we exposed them to this bacterial coat protein, lipopolysaccharide, at 1 of 2 times a day. One cohort got them at the beginning of the day, which is when mice become transitioned from being active to inactive. And another cohort got them at the end of the day, which is when mice transition from being inactive to being active because they're nocturnal. And then we kept the mice in constant darkness for 3 days. And then we sampled them a day and a half later at various time points, 0, 6, 12, and 18. So we sampled them over a 24-hour period, starting a day and a half after the treatment. And these are this is the result that you see. This is the number of neutrophils that we can recover from the lung in bronchial alveolar lavage, which is we can wash the neutrophils out of the lung from the mice after they're dead. And you can see that there's a really sharp peak response at this transition point here from activity to inactivity. And it's around about 2 to 3-fold higher than it is in the middle or late inactive period. And we've done this type of experiment dozens of times, and we always get this sort of 3, even 4-fold difference in amplitude of effect, which is strongly rhythmic and driven, as you're going to see in a minute, by the circadian clockwork itself within those epithelial cells. So here are the club cells that I told you about earlier. Here they are oscillating in culture. That's what they look like close up on a scanning EM. You can see they're surrounded by ciliated cells. And there they are lining the bronchioles. And this shows protein staining in a cross-section for the clock protein, which is one of the major drivers of the circadian clock, and another one down here, reverb here. And this clock protein almost exclusively co-expresses with a club cell marker, CCSP protein, and you can see the merge at the bottom there, merge between the green and the red. And if you look at that, you can see that those cells, pretty well all the cells have got both proteins expressed within them. So these are really strong candidate circadian pacemaker cells within the lung itself. We know a certain amount about the genetics of those cells, and for those of you that aren't familiar with these sorts of processes, it's relatively straightforward these days to genetically target these cells and pick on a gene of interest. So what we did was to knock the clock out just in those cells. So it's just one cell type in the whole body, and it's highly selective. That's what the little blot here shows. I won't run through the details of it, but specialists in the audience will probably detect what's going on. And it's highly selective, and by so doing, we were able to convert the lung from a rhythmic tissue, which is shown over here on the right, to an arrhythmic tissue. And when we take it in culture and look at how the lung behaves in culture, you can see that it's completely flat line. There's no rhythm in it whatsoever. And if you look for expression of the clock protein, you can't, the clock gene rather, this is an in situ hybridization, you can't see any of this gene in the lung at all. Whereas in the normal untargeted mice, which are on the left-hand side, you can see the bronchial structures have got very strong staining for this clock protein, and indeed another one down here at the bottom. So essentially what we've done is that we've killed the clock in just one cell type in the lung. And we can now go and ask what happens if we challenge these mice with a bacterial endotoxin, what happens to their response? And really, it was quite remarkable. You can see at the top here that the neutrophilic response, which is each of these little, I'm so sorry, I've got a very unstable computer here. You can see that we've got each of those cells, each of those little blobs there is a neutrophil, and this is a spread collected from the lung. And you can see that in the cells in which we've targeted the clock, that's BMAL1 there, we've got a very large number of neutrophils in response to an inflammatory challenge compared to the wild types. And at the bottom here, you can see that the numbers are absolutely overwhelming. You know, a massive augmentation of effect compared to the wild types. And this is graphed out at the bottom. And what we did here was to treat mice with this bacterial stimulus. And you can see that the time course of response, a single treatment, was augmented over a period of 2 or more days. In fact, we can still see significant differences at 3 days between the two groups, the blue being the clock knockout in those cells. And this shows some of the histology of the tissue. This is a wild type on the left-hand side, and that's the CCSP targeted mouse. And if you look close up here, you can see massive inflammatory responses in the lung here. It's a very sick piece of tissue compared to the normal wild type mouse here. And that shows the quantification, a bit more detail of the neutrophilic response. So we can say that the lung depends absolutely critically on an intact functioning clock in order to be able to show a normal response to a bacterial challenge and mount an appropriate neutrophilic response. And I'll talk a little later about some of the great dangers of shift work and messing with our own internal clocks. And we think an awful lot of the pathology and physiology that we see in humans that have got difficulties associated with shift work are underpinned by an augmented inflammatory response and chronic background inflammation across the body generated by long-term mistiming events. Just to finish this story off, we got very lucky because we screened for literally hundreds of candidate proteins that might be driven by the clock in those cells and that might be driving that, that process. And we identified a particular chemical signal, a chemokine, which was produced specifically in these cells of the lung. And it's called CXCL5. It doesn't really matter what it is, but we found one chemokine, and we're able to show when we knocked this out in mice, we could knock out the clock effect, so to speak. We could phenocopy the effect of the clock mutation by knocking that gene out. So it looks now that these cells here, these important bronchial epithelial cells, have got a really important role to play in the lung, timing immune responses, generating signals, in this case, in the case of the mouse, CXCL5, that then bring about this neutrophilic invasion that then deals with pathogens that are attacking the lung. And that's just a little summary of the story in mice. On the left, you've got a healthy mouse, which is not clock disrupted. And if you treat it with bacterial infection, you get a different response depending on the time of day. In the that's CXCL5 there, and that's at night when CXCL5 is down. And if you disrupt it by genetic mechanisms or even keeping mice on abnormal light cycles, you can generate this kind of pathology where you see augmented responses, major neutrophilic invasion, tissue damage, and serious pathology arising primarily from a disruption of the timing mechanisms that are driving those cells in the lung. And my colleague, Jeff Haspel in Washington University in the States, has picked this up very nicely. And I'll show you one more bit of work he's done that's just come out this month in a few minutes. And he's a physician with an interest in lung pathology and the clock. And he's recently pulled together a very clever analysis in which he's looked at all the published data on mice, including our own, and all the published data on human lung function. And the general conclusion from this is that pretty well all the genes in the lung that are under rhythmic control are in one way or another involved in immune function. Its primary role of the circadian clock in the lung is to time and tune the immune system. And in both mice and humans, the immune pathways are predominantly enriched at the gene expression level when those organisms are at their respective rest phase. So we are, as diurnal animals, going into a period of enrichment of our lung immune system at the gene expression level, whereas mice are just coming out of it because they're nocturnal. And I'm not going to go into the details of this. These are some of the circuits and gene pathways that are under control of the clock in the mouse on the right-hand side and in the human on the left-hand side. So we think this is potentially really important and has got all sorts of implications, one of which I'll tell you about shortly for human pathology. So just moving forwards now, I want to tell you a little bit about metabolic cues and metabolic links, because we've come to realize that this circadian clockwork that I've told you about, and this, by the way, at the top, I think it works, is a little movie clip of a mouse suprachiasmatic nucleus. There's the pair of them there. It's a brain section, a brain slice in culture. And this is from a mouse in which we've tagged one of the genes to a light-emitting luciferase, so we can measure light emission from that tissue in culture. And it will continue to rhythmically oscillate for many, many weeks and months in a nice synchronized pattern in that brain slice. But we've come to realize that the major component that regulates synchronicity of all of these peripheral clocks is actually driven through brain activity driving feeding centers in the brain, so rhythmic feeding. And this leads to metabolic synchrony, particularly in the lung and in the liver. And the synchrony of these peripheral clocks is very strongly controlled by the time of feeding. And we can demonstrate that quite easily in mice. I can just take this one forward. Okay. Here's one example for you. I've just gone through a story showing you in mice how the lung clock is very important in terms of generating the response to inflammatory stimuli. And if you look at the bottom right, panels F and G, you see the black data there. This is a replication of what I've shown you already. This is the neutrophilic response to LPS treatment in mice. And you can see you've got a really massive, sorry, it's terribly unstable, this, a massive response at ZT0 and very low at ZT12. And there's CXCL5, which is that chemokine I told you about, which is under strong clock control within the lung. And if we take a cohort of mice and keep them in a light-dark cycle, but don't allow them to eat at night when they would normally like to eat, but actually put the food in at the beginning of the day and take it out at the end of the day. So they're forced to feed at the wrong circadian phase, but don't forget they're still kept in a light-dark cycle. So the brain clock is still driven by the light, but the body clocks below the neck throughout the rest of the body are now driven by the feeding cue. And when we do that, we can completely phase reverse the inflammatory response in the lung. I'm so annoyed about this. I do apologize. And you can see that the neutrophilic response shown here is now at the lowest point at the transition to light and at the highest point at the transition to dark, a complete mirror image of what you normally see if a mouse is allowed to eat when it would like to eat during the nighttime. And this is true for some clock genes up here, which is what the top panels show, and that's true for total cellular infiltrate in the lung, which is essentially a replication of the neutrophil data. So what you're seeing there is very clear evidence that the immune function of the lung is really indirectly regulated through feeding. And the feeding is indirectly regulated through activity from the brain that controls feed centers, which is indirectly regulated by the impact of light acting through the suprachiasmatic nucleus. So it's like a whole row of dominoes going down. And the discovery of the role of metabolic cues and feeding has been enormously valuable because it's allowed us now to put structures such as the liver at different phases to the brain and ask all sorts of questions to do with what's the impact of feeding at the wrong time of day in animals. And more recently, these studies have now been done, of course, extensively in humans. So this leads us to the question as to, does it matter what time of day you eat? And it turns out it really does matter. And the initial studies, again, were done in mice. And at the top of this is just a cartoon summary of a whole series of experiments and publications. The top part of this figure shows an obese mouse model. This is a mouse that is genetically obese. So it's got a defect in some of the circuitry that controls feeding behavior. And if you allow an obese mouse to feed ad lib, it will become morbidly obese over the course of the next few months of life, which is shown right at the top there. But if you time restrict that mouse so that it can only eat for a specific time during the nocturnal period, the bottom there, that mouse loses weight. It's still obese, but it's much fitter, and you can assess that in a variety of ways, including wheel running activity, which is a nice, easy surrogate for it. And I should stress that in these experiments, the mice are controlled for how much they can eat. It isn't that the time-restricted mice are also forced to eat less food. It's just that they have to eat that food at a specific phase of the circadian cycle. You do the experiment slightly differently, and you take a lean mouse and you put it on an obesogenic diet, which is a high-fat diet for a mouse, and you allow it ad libitum access. It will get fat. By the way, mice get fat remarkably slowly compared to humans. They're quite good at resisting high-fat diets, but nonetheless, a mouse kept on an artificial high-fat diet will eventually end up obese. And if you give it the same high-fat diet, but you restrict the time of day when it's allowed to eat, so that it eats the same amount, but it's only specific to the circadian nocturnal activity period, it remains lean and fit. And you can even mimic human activity at the bottom in another study where people have taken time-restricted protocols in the work days and then given the mouse the weekend off, and it still remains lean and fit. So that tells us that the liver clock in particular is really critical in this, and that the phase of the cycle that the liver clock is exposed to nutrient is really crucial in terms of determining the metabolic outcomes. And more recently, colleagues have gone on to look at the interplay between clocks, caloric intake, and life expectancy. And there's a well-known effect that you'll be probably all familiar with, which is that consistent caloric restriction, low energy intake, extends life and has a positive impact on all sorts of potential pathologies, including diabetes, coronary vascular disease, hypertension, neurodegenerative disease, and inflammation. And it's believed actually that inflammation underpins an awful lot of those other pathologies. And this effect of calorie restriction has been reported in a wide variety of organisms, including yeasts, worms, flies, mice, rats. It should be mole rat there, not mole, and primates. It's less established in humans, but I think we have to accept that it's very likely. And there are different types of manipulation that you can undertake: calorie restriction, or time-restricted feeding, which is shown over here on the right. And that's been much less well explored compared to the general effects of calorie restriction. So the question now is whether these effects of calorie restriction that affect in a beneficial way longevity and pathology are interconnected in some way to the circadian clock itself. And the first hint of that comes from studies which show that processes that are associated with dietary restriction, and here are some of the pathways on this figure. Again, it doesn't really matter in too much detail as to what those specific pathways do or contribute to. But some are involved in pro-aging, and some are involved in anti-aging, shown in red and green respectively on the right-hand side. And what you can see there is a screen of different tissues. Again, this is mouse data, from the brain through to the liver, white adipose tissue, brown adipose tissue, and muscle. And you can see that large numbers of these pathways or genes are actually under tight circadian control in some or all of these tissues. Now, this is telling us that the pathways and genetics that have been identified by a separate group of biologists who are interested in the biology of aging have inadvertently stumbled over a subset of genes that we now know are also very tightly under the control of the circadian clock. So the trick now is to try and disentangle these two processes: the effect of dietary restriction on aging and some of the genetics that underpins it, and how the clock might be involved. And this is just one slide to summarize a beautiful recent study by a friend of mine in the States who undertook a gargantuan 3-year study in mice, in which he looked at the interaction between the circadian clock and calorie restriction on the aging process. And on the left-hand side, that's the top left there, you can see a mouse that is allowed to eat what it wants during the light-dark cycle. And mice, although they're nocturnal, will still eat a bit during the day, and that's what's shown with that little cartoon with the pellets. And they measured over the course of the lifetime of cohorts of mice circadian amplitude measured using telemetry and behavior and other processes. And you can see that as the mouse ages, the amplitude of the circadian clockwork diminishes. And that's true for us as well as humans. That's been very well characterized. And associated with this is increased insulin resistance, which is shown in that little cartoon there. You can see the little symbol showing the increase in insulin resistance as this mouse ages there. In the middle panel, you've got the effect of calorie restriction, and this is using a model in which mice are fed through an electronic hopper system that delivers one pellet every hour or so, and they're hungry because they eat that because they're only given 70% of what they'd like to normally eat. And that pellet is distributed evenly over the course of the light-dark cycle, shown in the middle there. And that leads to about a 10 or more percent, 10 to 15% increase in longevity. And that is typically the sorts of life extension that large numbers of biologists working in the field of aging have uncovered. It's about a 10% effect. And you can see that it has a slightly beneficial effect on circadian amplitude, a little bit more peaky there, and a beneficial effect on insulin resistance, shown just below that. But if you do a really interesting experiment and you combine calorie restriction with time of day restriction, so you allow the mouse to eat 70% of what it wants to eat, but only during its normal active phase at night, and you don't spread it out during the day, you get a massive effect on circadian amplitude, and you double or treble the increase in longevity up to 33% in this particular set of examples, and improve insulin resistance. And all of that's associated with beneficial effects on endothelial health, glucose uptake, muscle growth, triglyceride synthesis. I'm summarizing a really complicated series of experiments with massive amounts of data behind it, but those are the sorts of systems that are impacted. So the implications for us are really clear, which is that both diet plus controlling the time of day when you eat, the interaction between those two systems has a massive potential benefit. And in fact, diet alone is overwhelmed by the effect of the circadian clock. If you can add the clock knowledge and clock logic to the process on top of diet, you get an enormous increase in benefit. That's the take-home message from that study. And this has led now to an interest in circadian medicine and how circadian rhythms may be important in the aging process. And shown at the top, you've got a little cartoon that shows a young to an aged organism. It could be a human, it could be a mouse. And the ad lib group, shown in red, sorry, the colors are reversed at the top. You get a diminishing amplitude of the clock, whereas in the calorie restricted group, shown in green, you get an augmented effect. Eat less, take more exercise, and it'll have an extraordinary unexpected effect on your circadian clockwork. And a number of groups, including ours, have become very interested in the potential of small molecule activators and regulators of those clock proteins, and whether circadian mimetics can actually impact on the process of aging itself. And the question now is whether these drugs are best administered in phase with the circadian clock. So shown at the bottom there is a little model for you, the need to tailor the drug to your optimal time, in which you restore to a youthful state your organism or yourself if you tailor it to the optimal time compared to the suboptimal time. And if all that sounds like a lot of arm waving, just wait a minute, because I'm going to show you some data from real drug trials and real studies in humans, and you'll perhaps get to see the scale of the problem and also the challenge that lies ahead in terms of how we translate these data into human benefit. So just to finish off, I'm going to say something about the sleep cycle, and we'll come back to drugs in a minute. And this is my friend and colleague, David Ray, who worked with me for many years at Manchester and has now moved to Oxford, where he is the professor of endocrinology. And he's particularly interested in the circadian clock. And the question here is, are we facing an epidemic of exhaustion? And here are some statistics for you. This is from a study in the States. 30% of all Americans sleep less than 6 hours per 24 hours, and 44% of night shift workers sleep less. 50 years ago, it's thought that this was only about 3%. So we've undertaken a massive change in our own life cycle. We've adopted a 24-hour life cycle for many of us. We've got more light in our lives, we sleep less. And in the US, they reckon between 50 to 70 million people suffer some adverse health consequences, some sleep disorders and deficiencies. And here's an extreme example for you. Here are these 4 catastrophes: major airline crash, Exxon Valdez oil tanker spill, for those of you that can remember it, 3 Mile Island nuclear disaster, and of course Chernobyl. What's the link? The link is these were all associated with sleep deprivation and lack of sleep. Exxon Valdez, the tanker, the captain of the tanker, had apparently had no proper sleep for 3 days prior to the accident. And the accidents in the 2 nuclear events at the bottom were both on the night shift with sleep-deprived staff. So the sleep process that drives physiology is underpinned by an underlying rhythmic circadian clockwork shown in the black at the bottom here, and the need to dissipate events associated with so-called process S, which rises during the wake phase and is then dissipated during sleep. And as a consequence of this, if you're sleep deprived, you accumulate more of process S. I'm not going to go into any details to what that actually means in terms of biochemistry, but those circuits are now being dissected. And that's actually driven by a gating mechanism, which is the underlying circadian clock control that regulates sleep. This explains why, even though we're very severely sleep deprived, it's sometimes very, very difficult to actually get restorative sleep if we're trying to sleep at the wrong phase of our cycle. And fatigue is rather like being drunk. This is, again, it's a US study, and this compares the effect of fatigue and wakefulness with blood alcohol content. And I'm not going to go into the details of all these stats, but essentially, 1 night without sleep is equivalent to being over the legal limit in terms of your reaction time in a variety of different tests here compared to alcohol. And so the question is, would you like to be driven by a drunk airline pilot? Well, the blunt truth is that probably many of us have been, not drunk, but certainly severely sleep deprived. So shift work has a major impact on health. It drives misalignment of those clocks, those peripheral clocks that I was telling you about. It's associated with liver fat accumulation, and the liver fat is linked to insulin resistance. And this can be tested directly. And I think because we're now approaching 1 hour, I'm going to skip the next 2 slides and move on to the very last bit. And I'll come back to those 2 slides if anybody's interested. I do apologize for the overrun. But this is just the last bit of it, which is brings us back to the immune system. And the idea that the clockwork that regulates immunity in animals is critical in us as well. And some of the evidence of this has come from studies of vaccination. And in particular, comparing the efficacy of vaccination protocols administered at different times of the day. And the first major paper was from Janet Loudon colleagues at Birmingham, which was published in Vaccine a few years ago now. What they did was to compare the antibody responses of people to influenza vaccine in the morning compared to the afternoon, and showed that there was a significant enhancement of the antibody response if the vaccination was given in the morning compared to the afternoon time of day. It was a relatively underpowered study, and parts of it have been slightly revised, but the same message still stands. But it wasn't a really big study, and although it got a lot of publicity, it suffered, as many circadian studies do, from the fact that it's very difficult to get good, strong circadian.
I'll have a cup of tea.
Hello, someone there? Okay. Then what emerged?
I think somebody has unmuted.
It's okay, don't worry. We're nearly there. So what then emerged was some evidence. These are from 3 or 4 separate studies. This is a review paper that was published a year or 2 back now, that showed that for a number of vaccine protocols, including COVID-19, the efficacy of the humoral response that was measured in people was directly linked to whether they were able to establish good sleep patterns after the vaccination. And this led to the idea that a good night's sleep after vaccination had a beneficial effect in terms of your ability to mount a robust immunological response. And I mentioned my friend Jeff Haspel. I showed you that comparison between mouse and human lung earlier on in the talk. And he's just published, it came out today, actually. I knew it was on its way, but this is the paper in Journal of Clinical Investigation. It's only available online. And what Jeff has done is he managed to get access to an enormous data set from Israel. And they looked at a total of 1.5 million people who'd received COVID vaccination. And these all had a time signature on them. So they were able to assess the efficacy of the vaccination when the vaccine was given at different times of the day. And one of the parameters that they measured was breakthrough infections, people who subsequently developed COVID despite having had a vaccination, and looked at that as one of the parameters. And the message here is very clear. Breakthrough infections differed based on vaccination time, with lowest rates associated with late morning to early afternoon and highest rates with evening vaccination. Exactly the same time points, by the way, that the earlier study on influenza also showed. So it looks to me like if you're going to your doctor to get a vaccination, try and get a mid-morning appointment. Don't accept a late evening appointment. I've done that to my own GP, and my advice to you listening is to do everything that you can, if you're going to be vaccinated, to make sure that it occurs in the around about the middle of the day and not later in the day or in the evening. This study here is the best I've seen so far, and it's a massive study with very large data sets, and there's a very complicated set of parameters that are measured in the depth of the body of the paper if anybody is interested in following it up. But it's a really fine piece of work. So just to finish, I think this is almost the last slide. This is a study that was published by colleagues of mine in Cincinnati. And what they did was to look at a total of 106 different human trials in which they had time data available for the drug. And they were able to assess that of the 106 trials tested, these are different drugs. 78 of them, the great majority of them, had a time of day effect. The efficacy was time of day dependent, and that's That's what's shown on the left-hand side. That's a date line from 1970 to 2020. The blue are those that have a time of day signature, and the trial size on the left-hand side is shown on a logarithmic scale. And in the middle there, you've got the types of drugs that are being tested: hypertensive drugs, cancer, asthma, and so on, allergic and other drugs. And then on the right-hand side, you've got data on the known half-lives of these drugs. And you'll see that the majority of them have got relatively short half-lives. Now, this is a really interesting question, because if you talk to people in the pharmaceutical industry, and I've worked with colleagues in GSK, one of their concerns are to develop drugs with relatively long half-lives so that they can have a persistent effect over a 24-hour period. But if you're trying to hit a specific circadian target at a specific phase of a cycle, you actually want to re-engineer quite a large number of those drugs so that they become slightly destabilized with a short half-life. And it turns out that the drugs that had the strongest time of day effect are those that tended to have the shortest half-lives, which would fit really, because if it had a really long half-life of more than 24 hours, you couldn't, by definition, easily see a time of day effect, because the drug would still be around over the course of the next circadian cycle. So for drugs and drug usage, the circadian clock really does matter. And it's turning out that a very large number of drugs are known to hit circadian targets. And therefore, the time of day that those drugs are administered is likely to be critically important. If you ask your average medical school in this country how much of this information is permeated into the teaching of the current cohorts of medical students, the answer is pretty well none. So all of this is yet to come, and yet to get into an already very crowded curriculum in terms of how we teach and train our health professionals in this country and elsewhere. So the implications for chronotherapy, and I think this is the last slide, are considerable. I've just talked about immunological responses with a little bit of a side talk on aging and metabolism. There are vast numbers of other areas. I nearly hardly touched the topic of sleep, for instance. But many of the processes that are common and of concern to medical professionals are amenable to chronotherapeutic treatment. And although they don't offer a binary cure in the sense that you can actually get rid of a process, they do offer the potential for a considerable reduction in pathology, in clinical load, and often, particularly in the case of processes such as asthma, it may well be possible to improve treatments to the extent that people who are simply disabled and can't work are able to get back to work and a normal life as a result. So it's early days. The number of really well conducted clinical trials are thin on the ground. They're very expensive to put together. The money's tight. And colleagues of mine in the clinical area are working hard to try and rectify that. But I'm hopeful that over the course of the next 15 to 20 years, we're going to see some of this science permeating into general medical practice, and of course, being generally better understood by the population at large. Because if people don't understand the importance of their own clockwork, there's very little point in a doctor telling you to do something at a particular phase if you just ignore it and pay no attention to it. So there's also a major public understanding of science job to be done in this whole area to try and get over some of these quite complicated studies in a simple way, so the general population is better informed. there's been a very large group of us working on this. I set up the timing group at Manchester, which is now one of the largest in the world. And just on the left there at the top, it's a little subset of people who've just been involved in the immune work. There are lots of other people doing other types of things. I retain very strong links with David Ray and colleagues at Oxford. And we've collaborated in some of the studies I've told you about with friends in the States, in Harvard, Massachusetts, Colorado, and Oregon. And also, it's really important professionally to thank and recognize the funding agencies who kept us going over these years, particularly the Wellcome Trust, and then the Medical Research Council and the BBSRC. And in the early days, GSK, and hats off to them. They were one of the first major pharmaceutical companies who saw the importance of this and went into this in a big way 15, 20 years ago and supported a chronotherapy initiative at the University of Manchester. And I'm very grateful to them for that seed funding.